Hello, everyone. Today we have Martin Holmes, who is a junior at Caltech studying bioengineering, and we're just going to go over like some of the research that he's done, as well as some of the classes and the Caltech experience that he's been through, and just give you guys a little bit of an insight to what that is. All right, take it away.
Okay, hi everyone. I'm Martin. I'm Mason's brother, and I'm studying bioengineering at Caltech. And I just wanted to show you what being a bioengineer in college kind of looks like and what you can expect to study. I do a lot of classes in terms, and I wanted to show you kind of the stuff that you would expect in classes. And I'm a junior right now, so this would be your third year classes. And I wanted to show you some of the research I do on the side. So you can expect how to like kind of what kind of things you'd be introduced to. So I'll start a little bit with my research that I did over the summer. So I'm actually moving away from this kind of research, but I came into Caltech expecting to do a lot more gene circuits and synthetic biology as a bioengineer. So the first lab that I got into freshman year was this lab. So what they do, or what my project was, is E. coli genome minimization. And they use this cool new technique, rexogenesis. The technique itself contains a lot of buzzwords, and I was just going to go over a little preview of kind of background about what kind of things that I study. So, not here. Okay. So I'm basically, the goal of the whole project is to minimize the E. coli genome. And you might ask, like, why would you want to minimize an organism that already does its functions properly? Well, it's basically a proof of concept. We're doing it as to show that we can as an engineering principle. And there's some small applications that can actually be used in like industries and stuff, or in other research. Basically, mostly about combinations of lethal genes inside genomes, as well as creating a clearer environment. If you're minimizing a genome, you don't produce as many proteins, and that clears up kind of all the metabolites that or metabolic proteins that E. coli can have. So you can do better studies in the future and have less proteins being influencing that. Also, it just has a general understanding of like what genes are necessary for survival of microorganisms, and how that basically plays into life. So genome minimization has been around for a little bit, but there's been limited success, and it doesn't always involve model organisms that we use often in synthetic biology. So if any of you are familiar with some synthetic biology, you know that E. coli is a really big organism that engineers use. And so it's very well studied. And that's the reason why we're using it in this minimization project, because we know a lot of those genes that and what they do. So we know what we can delete, what we can't delete, and what the meaning of deleting one of those genes is. Yeah, let me see. Sorry, my computer's having a little bit of trouble. So the first thing that you have to do when you're minimizing any type of genome is you have to go and look at all the essential genes that genome contains. And scientists before us, research scientists, go in and look at the genome and they screen. So basically what they do is they take all the genes in the genome and they make certain deletes or certain interruptions in the genome in specific places, and they see if the bacteria can survive. That seems like kind of random and a kind of like a long process, and it is. But they do these screening tests to look for all the genes that are necessary for survival. And you can see that how many genes you can actually delete within on the right here on the genome. You can see how many genes you can delete before the bacteria that the bacteria can still survive on. So we identify regions that we want to keep, and actually I'll show you later kind of the mechanisms that we use to keep these regions. What we actually do is we reintroduce the keep regions and take out the original genome sequentially to perform this. So the way that this buzzwords come into play when doing this minimization research and rexogenesis is we have CRISPR, which is a big word right now, as well as lambda red type homology repair. And we use these basic properties or basic tools to go through the genome and recreate a new minimized genome. So as you can see here on the diagrams on the right, we make synthetic chromosomes, basically synthetic large DNA fragments, and that contain only the regions that we want. And what we do is we introduce them into an E. coli cell and use CRISPR and lambda red to reintroduce those synthetic fragments that only contain the keeps into the entire genome where those keeps used to reside. And you can do this sequentially and basically re-engineer the entire genome. This has a lot more applications than just minimizing it, but it's a really cool method that is pretty novel, that you can recode entire prokaryotic genomes with. Other people in the lab do things like make something called non-canonical amino acids, basically amino acids that we don't see in nature, and they recode entire proteins and areas of the genome crazy ways. So this is a pretty powerful method that we can go into, and it just takes some time to go in and identify all the key regions, put them together, repair things, and in the end, I'm not going to show you all these things, but in the end, you only need a few things to actually create a minimized genome. But it just takes a lot of design principles. And so that's kind of the research that I do outside of the class. And I just wanted to show you kind of what my term this year. So Caltech works where we have 3 terms and then a summer like kind of session where we can do research on campus or on other campuses. So I just want to show you what this portion of the year looks like, my my fall term, so that you guys can expect like what you would do if you're a bioengineering student in college. So my current schedule, you'll see actually that bioengineering is really cool, and that there's a lot of interdisciplinary engineering and even sciences that go into your course load and what you have to learn. So the first class, I'll show you examples of what I do in each of these classes in the next few slides, but I'm taking 4 courses this term. The first one is a biodevices course. As you can see, it's co-labeled with electrical engineering and medical engineering. And I'll show you kind of like what I have to build. We get to build circuits. It's really cool. The next one is biomolecular engineering and the introduction to it. It's a chemical engineering course and as well. And it's actually taught, or used to be taught. This year she's working with the president. But it used to be taught by a Nobel laureate who has really cool protein engineering kind of abilities. She just won, that's why I mentioned it in the last, I think it was like 3 years ago. And then I'm taking some introduction, some big CS. What we'll see is a lot of my courses are CS based. And I'm taking organic chemistry a little bit late, but I just wanted to show you. So as a bioengineer, some of the things you have to do are build circuits. This is a circuit I built recently for my class. It has some messy wiring because I was late to turn it in. But this is like an example of a filter that I built.
Wait, do you mind switching over to the other presentation?
Sorry?
Do you mind switching over to the other presentation real quick?
I am on the other presentation. Oh, it's not showing?
Wait. Do you mind switching over? Sorry.
It says my screen sharing is paused. So I am on the other presentation. Do you not see, when I was speaking, did you not see my presentation? Oops. I think
it got stuck on one of the components slide.
Okay, let me just switch that really fast. Can you see it now?
Yes, perfect. Thank you.
Okay, yeah. So here you can just slice that out. So here we have Caltech's bioengineering. I'll go over again. I already just explained these my course schedule. I have a combination, just to review, I have a combination of a lot of different types of engineering and sciences that go into what your course load is in a term for bioengineering. So here's the circuit I was discussing. As you can see, the wiring is still messy. It didn't change from the last time I just presented. But I was filtering some data that was sent to me. This is a band pass filter. It has a high pass and low pass. Right now I'm actually building a pulse oximeter for that class. That's the thing that you go to the doctors and you put on your finger and they measure your heart and oxygen levels and stuff like that. So I'm still working on that right now, but it's actually a really cool class. This is a basic circuit, but I think things will get, we use a lot of different components to do a lot of different things. Another class I'm taking is biomolecular engineering. This is straight from a problem set that I'm doing right now. So you can expect lots of basically DNA machines and DNA computing and DNA building. So the diagram on the left here is basically a reaction mechanism to build the center. I don't know if you can see can you see my mouse? The center structure right here. And what you can do is, you know what structures you want to build. You can actually run, I do a lot of coding for this class. You run programs and sometimes code up from scratch those programs, or almost scratch. Basically the assembly process, picking what nucleotides you have to choose to create these structures, predict those secondary structures, and then combine them. So there's multiple strands that go into creating these big structures, and it's really cool class. On the right here is a simple logic gate that I made recently. It's basically this is an and or gate. So if X and Y exist or Z exist, you'll have a fluorescence in your tube. I'm also taking an introduction to data analysis biological sciences. It's a statistics CS based class. So it's a lot of like looking at big data sets. The one on the left was something with Gal4UAS, which is like a protein they use often genetic system for screening and stuff. So, and on the right is actually not from this class, but I wanted to show, it was from, I think I took it my freshman year, but this is like an example of filtering data that you can use with this class, but it wasn't specifically in this problem set that I did. And last one is organic chemistry. I'm sure you all are familiar. This is a chair flipping around, but it's just basic for now. But you have to know it when you're in bioengineering, because you have to know biochemistry. But yeah, that's just an overview of like what I'm taking this term and the research that I'm doing right now. So yeah, I'm excited to share that with you guys.
So we actually do have a question.
Yeah.
Let me pull it up. Okay, the question is, what is like the academic pressure at Caltech, and how manageable is it as a first year? And like, do you have free time?
Actually, so first year is really cool. Right now, I don't have that much free time. Actually, I'm mostly excited about the courses that I'm taking. And it's enjoyable in that way. But first year is actually really cool. So for the first two terms, so there's 3 terms in a year, first 2 terms, all your classes are in pass fail, and all the freshmen are taking the same classes because there's a lot of core curriculum that you have to take, a lot of math and physics, and something that as a bioengineer, I have to take a lot of physics as well. So you have to take a lot of those classes. It's actually really fun. You're on pass fail, so there's a lot of like adjustment period. You have a lot of time. I had so much fun my freshman year to meet people, and the classes are pretty cool. Right now, as you can see, I'm very excited about my classes. So I don't have that much free time at all junior year. It's just how I planned my schedule, because I chose to take a lot of classes. But yeah, the academic pressure is not bad at Caltech. I went to the same high school as my brother, but there's not the sense of competitiveness at all. People are always willing to help you on problem sets. Like, for example, sometimes I need help understanding something from organic chemistry. Sometimes I need help debugging my code in my classes. And my friends and people I barely even know are always willing to come and help. Like, I've met so many people from my class because I asked them a question, and they're always available to help and collaborate. Basically every single class, all the sets are collaborative. That means that you are encouraged to work with other people on the set. And in fact, my coding class, does it work? Yeah, this coding class is group-based. So I work with people and turn into my sets with them together. So yeah, there's not that much competitiveness. There's just a lot of things that you're able to learn. Yeah.
All right, we have a question from Chris Jung, and it reads, what would you say your typical schedule is, and how close can you get to your professors? How different is the learning process specifically from high school?
Okay, yeah, that's a good question. So I kind of, what was the first part of that question? Do you mind?
The first part was, what is your typical schedule?
Oh yeah, so I just went over my schedule. So I do a little bit of research. That was mostly over the summer and a little bit of the beginning of this year. And then I went over my schedule with my classes if you just came in. And I like build circuits, lots of everything from electrical engineering to chemical engineering, some chemical engineering, building DNA circuits, and not like logic gates and assembling machines and structures, and a lot of CS based stuff. Every year, every term is a little bit different. If you're asking about like freshman year, you take a lot of freshman year, you take like basic chemistry, math, physics, all your good stuff for that year, and you get to work with everybody. So it actually is very enjoyable. I had a lot of fun freshman year in those classes because it's all collaborative. So you're working with your friends on your sets, and it's kind of fun to play around with like the ideas of what you're working on. And Caltech is a really small school. There's less than 1,000 undergraduates. So all three of the classes, the electrical engineering, chemical engineering, bioengineering, all those three first classes that I showed, they're all with PhD students because they take the same classes as us, which is really cool. So a lot of like, for example, my group members in my data analysis class are PhD students. And you get close to your professors because it's a very small school. You can choose how close you want to get with your professors, but I got close with one of my professors this year because I'm in a lot of his classes, and it's nice. You get to know them. Sometimes it's a little bit of a curse because they know you so well, they look for you in class, and they make sure that you're doing all your work. So it's nice. It's much better than high school where it's a little detached. Also, college is largely application based. So I'm doing a lot of hands-on, as you can see, building circuits, a lot of computing like lots of things. So high school is not a fun time. You don't get to do as much. The learning is mostly memorization. And in college, there is memorization, but a lot of it is the process, which is really cool.
Are there any other questions? Okay, if that's it, then I think that we're gonna end it here unless there's anything else that anyone wants to add. Thank you so much for everybody who came, and thank you so much, Martin, for hosting this webinar.
Yeah, of course. All right, thank you, everyone.